Good morning, welcome. My name is Emily Martin, and I am pleased to join you today in my role as Clinical Affairs Specialist at Fermimpex NV, the distributor of bioMérieux diagnostic solutions for the Caribbean. At Fermimpex, we are dedicated to advances healthcare through innovative solutions and meaningful knowledge sharing. When people and institutions come together to contribute their core expertise toward a common goal, it creates an environment where success is inevitable. I sincerely thank you for being and for joining us for this event to launch the new VITEK 2 gram-negative cards for the Caribbean region. This event represents an important milestone within the VITEK system. As many of you know, bioMérieux is recognized for its leadership in developing sustainable, reliable, and innovative solutions for the diagnosis of infectious diseases. In this 1 hour and a half session, which will be divided into a main presentation and a Q&A segment, we will be joined by Professor Germán Esparza, who will share an overview of the new cards, their clinical implications, the key improvements, and their key impact in both hospital and outpatient settings. It is my pleasure to introduce to you our Caribbean friends, Professor Germán Esparza. Germán is a microbiology specialist. He serves as an expert panel member on the Microbiology and Antimicrobial Susceptibility Testing Subcommittee at the Clinical and Laboratory Standard Institute, CLSI, in the United States. He is also a member of the American Society for Microbiology and Antimicrobial Stewardship Committee at the American Infectious Diseases Association. Currently, Germán is the director of the proficiency testing program in clinical microbiology pro-ASIACAL. He works as a professor at the clinical microbiology and antimicrobials at Pontificia Universidad Javeriana in Bogotá, Colombia. Please join me in giving a warm welcome to our friend in the Caribbean, Professor Germán Esparza. With such an accomplished background, we are confident that this presentation will provide you valuable insights and inspire us all. Before we begin, just a few housekeeping notes. The webinar will run approximately 1 hour and a half with a dedicated Q&A session at the end. Please feel free submit your question at any time via the chat or the Q&A box. I will make sure they are addressed during the discussion segment. We hope this webinar will make a significant contribution to strengthening your knowledge and supporting better decision-making in clinical microbiology. Professor, the floor is yours.
Thank you very much, Emily. Good morning, everybody. It's my pleasure to be here. I would like to thank Fermimpex and the International Division from bioMérieux for this invite, and all of you for attending this meeting. As was mentioned by Emily, we are going to talk about enhancing gram-negative AST with new VITEK 2 cards in the Caribbean. So welcome, everybody. This is my disclosure. I work as an advisor for the pharmaceutical companies developing antimicrobials for Latin America and the Caribbean. And I'm also an advisor for diagnostic manufacturers performing panels and cards for antimicrobial susceptibility testing and rapid diagnostic solution for antimicrobial resistance. But the concepts, copyrights, and opinions belong to me and do not represent any company here disclosed. So we are going to discuss the new VITEK 2 course for gram-negative bacteria in the Caribbean with some recommendations about implementation, expert rules, ancillary tests to detect clinically significant antimicrobial resistance mechanisms. As was mentioned by Emily, this webinar will be focused on gram-negative pathogens because unfortunately, this is our biggest problems of antimicrobial resistance in our region. So this is what we have for antimicrobial susceptibility testing methods so far. We have phenotypic methods like this diffusion, dilution, gradient diffusion, chromogenic media, or automated devices. We have also genotypic methods like PCR and several types of PCR and sequencing. And we have new methods coming to the market like microfluidics or ATP bioluminescence and many others coming to the market. But commonly used AST methods or antimicrobial susceptibility testing methods are quite different. For instance, we have qualitative methods like this diffusion that many of you have been using for many years. It provides categorical results like susceptible, intermediate, or resistant, but unfortunately, it doesn't provide information about how much resistant or how much susceptible the pathogen is, because we measure the inhibition zone diameters. Obviously, it has a range that we consult and we check this out in our standards like UCAS or the CLSI, but there is no relationship between the higher or the wider the zone diameter inhibition is, the susceptible the bacteria is. You cannot do that correlation. So you can just have a category with this result that is qualitative. But you have quantitative results providing the MIC of the or the minimal inhibitory concentration. So beside the categorical interpretation, the quantitative methods provide the MIC, which indicates how much susceptible or resistant the pathogen is for a particular drug. It is a better predictor for clinical outcomes. So let's put this in context. If, for instance, you have a Pseudomonas aeruginosa that is susceptible to cefepime, it's not the same that I call it susceptible with an MIC of 8, that is susceptible using our current breakpoints. But what about if this Pseudomonas is susceptible to cefepime with an MIC of 0.25 or 1? Is much more susceptible than the result having an MIC like 8. So it is important because it's a measure of how much resistant or susceptible the pathogen is. So what is the rationale to report MICs in our AST results? The MIC defines the required antibiotic exposure to guarantee that the patient can get the maximum clinical benefit. The MICs let you choose the antibiotic with the highest probability of clinical success. The MIC helps to monitor the presence of mechanisms of resistance, and this monitoring could be individually in a single AST report. For instance, when you perform AST for a patient having a urinary tract infection or intra-abdominal infection, or in the hospital antibiogram. That is something that other authors and other guidelines call like cumulative antibiogram, defining the most appropriate empiric therapy using the MIC distribution. So we have several methods to determine the MIC. We have the reference broad microdilution. Unfortunately, this method is not available to be performed in a clinical lab because it's cumbersome, it's difficult to perform, and it requires a lot of time and expertise to be done. Unfortunately, this is the reference method, and I think it's going to change in the future. But for most of the drugs that we are testing today, the reference broad is the one that we use to compare the commercial AST methods, but not for all the drugs. For example, for fosfomycin and mesalinam, we use agar dilution. Agar dilution is a method that uses agar that is melted, and we put the bacteria with a replicator over the surface, and we use to perform several dilutions of this drug trying to find out the MIC with this method. It's also cumbersome, it's not easy to perform, but as I mentioned earlier, this is the reference for fosfomycin or mesalamine. The two broad macro dilution, we use macro dilution for some drugs like colistin, and we have been using this for some disillusion methods. For instance, this synergy testing for ceftazidime avibactam plus astronam for metallocarbapenemases. This is easier, but it's also cumbersome in the way that we require a lot of tubes. And we require the dilutions of the drug, but it's a good method also. We have to use triplicates. The gradient diffusion that now we are using, like it tests for several new drugs, it provides also the MIC. It's easy to perform. It's very similar to this diffusion. You perform like a massive plating in a Merkinton agar, and you place the strip and you measure the ellipse or the inhibition zone here. Where you see that it cuts, you can get the MIC here for the result. And automated MIC methods like the VITEK 2 that we are going to talk about this today. So VITEK 2 is different. It provides dynamic and multidimensional MICs. The VITEK 2 continuously monitors growths in all wells. The growth in the positive control well is monitored until a predetermined minimum amount of bacterial growth is detected through turbidity measurements. The growth in the control well shows that the test isolate is viable and growing at an appropriate rate to begin analysis of the various drug wells. This analysis continues every 15 minutes until the susceptibility test completes. So this is a different way to do so. This is dynamic MIC, and it's very rapid. So with VITEK 2, you can get lower and adaptable MICs. Why lower is important? Because as lower the drug is for, or as the lower MIC for a specific antimicrobial drug is, it is important to predict clinical outcomes. Unfortunately, other automated systems available in several institutions have very fixed or very rigid, if you will, concentrations in the panels. With the VITEK 2, you can lower the breakpoints and lower the concentrations for a better detection of antimicrobial resistant mechanisms like ESBLs or carbapenemases. And for some drugs like ciprofloxacin, having lower MICs are useful to predict clinical outcomes for several infections. And the other thing that is important to mention with VITEK 2 is that it's fast. And the faster, the better. It's very rapid. So you can get, for instance, susceptibility results from 2 to 18 hours. In some institutions, having, for example, night shifts or afternoon shifts, they can provide AST results and also perform AST cards to be read next morning. So in that case, they are improving the turnaround time for specific wards, for instance, the ICUs. So let's talk about the new VITEK 2 cards for the Caribbean. So we are going to move card by card, and we are going to slowly explain what it means, what are the drugs meant for, and we are going to give some recommendations. And as was previously mentioned by Emily, we are going to have a Q&A session to exchange some thoughts with you about these cards. So why is required to update the AST cards? To improve the system performance, antibiotic formulations in the VITEK 2 cards need to be updated. With this improvement, the accuracy of MIC determination is preserved. For instance, colistin. Do you recall that colistin used to give some false susceptibility results? Now, in some cards, we have newer concentrations and newer formulations for this drug. And in a new card that you are going to see in some minutes, you are going to find out that we have polymyxin B. That is important for some infections, mostly caused by carbapenem-resistant Enterobacterialis, Pseudomonas, or Acinetobacter baumannii. The AST breakpoints are updated annually by the CLSI or the UCAS. New breakpoints may require different concentrations in the wells or different calling range. What I mean is the minimum or maximum values or MIC values to be detected. New antibiotics are coming to the market, and I have been visiting some of the islands and territories in the Caribbean. Probably I have met many of you here with me today, and it's really concerning that many territories in the Caribbean do not have the new drugs. And I think that in the way that carbaphenemases are moving around our countries, it is important to have availability of the new inhibitors, new beta-lactamase inhibitors for some patients. So we are struggling with that. We have been working with PAHO, with ICARS, and some other colleagues, with our colleagues at CARFA, because we definitely consider that this is a big deal, and this is very important for the Caribbean institutions. An emerging mechanism of resistance requires different or lower concentrations in antibiotic wells. For example, new beta-lactamases, like the carbaphenemases that we are facing. In some islands and some territories, for instance, I would say Trinidad, I would say Barbados, or I would say other islands in the Caribbean, we have been seeing carbapenem resistant Pseudomonas and Acinetobacter, but also KPC producing Enterobacterales. Unfortunately, it's coming to the Caribbean, this new beta-lactamase that is called the NDM, the New Delhi metallo-beta-lactamases. We have some reports in Jamaica, in Trinidad, in Barbados, and I'm concerned because this is the biggest public health threat that we are facing in Latin America, in South America, the NDM, also in Central and North America. So this is a different enzyme that requires a different approach. And device manufacturers prefer to have regional cards. It's very difficult to perform a country-based card because it requires time, it requires different antibiotic formulations, and this is very difficult to be done by the device manufacturers worldwide. So let's talk about the new VITEK 2 card AST 450. So this card contains amikacin. This is the low MIC, the high MIC, and the concentrations included. This is only inside the card. You are not supposed to see this in the report. Just the MIC, the calling range is this, but it will be adjusted to the current breakpoints. So we have amikacin, ampicillin sulbactam, cefazolin with a lower MIC. It is important this drug to de-escalate patients. For example, if your patient has been treated with ceftriaxone, you can test cefazolin, and if cefazolin is susceptible, you can de-escalate the treatment. And this is a decision that should be made by the attending physician, obviously. And also cefazolin is important to predict some oral cephalosporins, as I'm going to mention in a minute. We have here cefuroxime. This is a second generation drug that it has oral and IV presentation, IV formulation. So some institutions start the patients with the IV cefuroxime, then they switch to oral. This is what we call sequential therapy. And it could be important for some specific pathologies, for example, for prostatitis. We have here ceftazidime, we have ceftriaxone, we have cefepime, we have ciprofloxacin with the lower dilutions. It matches with the current CLSI and UCAS recommendations to test Salmonella and Shigella. We have ertapenem, the ESBL test, fosfomycin, gentamicin, meropenem, nitrofurantoin, and trimethoprim sulfa. As you may see, as you may notice already, this card is mostly for urinary tract infections, but could be used also for some other infections where oral alternatives are required to provide oral treatment. So which are the main features of this 450 card? This card is indicated for antimicrobial susceptibility testing of community-acquired infections caused by Enterobacterales only. This card is not intended to be used for Pseudomonas aeruginosa isolates. So those Enterobacterales may come from urine, stool, wounds, and other samples according to local guidelines. For instance, a wound, you can use this card to test trimethoprim sulfa, ciprofloxacin, and for UTIs, obviously, you can test for nitrofurantoin and other drugs. It also provides oral alternatives for patients with chronic infections. For example, osteomyelitis, those patients you know that they are going to have several weeks of treatment at home, or patients discharged for outpatient therapy. Ampicillin sulbactam susceptible results can be used to infer susceptible results to amoxicillin clavulanic acid. If some of your institutions use amoxicillin clavulanic acid for treatment, you can use a rule in the software that whenever you have E. coli or pneumoniae, which is susceptible to ampicillin sulbactam, the software can call amoxicillin clavulanic acid susceptible as well. Lower ciprofloxacin dilutions match the current CLSI and UCAT recommendations for testing Salmonella, and this card is also suitable to test Shigella and Stenotrophomonas maltophilia, where you require trimethoprim sulfa. VITEK 2GN450 incorporates lower dilutions for cefazolin. As I mentioned, this is the first generation cephalosporin used as a step-down therapy for infections caused by E. coli, pneumo, and Proteus mirabilis. Cefazolin is a surrogate to predict in vitro activity of oral cephalosporins like cephalexin for the treatment of uncomplicated urinary tract infections. This new card includes also cefuroxime, a second-generation cephalosporin suitable for sequential therapy IV to oral for selected infections caused by E. coli, pneumo, and Proteus mirabilis. So some recommendations when you consider to use VITEK 2 AST N450. This card replaces the VITEK 2 AST GN401, what is currently in phase out. So this card is not going to be available in the near future. This card is not intended to be used for Pseudomonas aeruginosa. We're going to have other options that I'm going to mention in a minute. Ampicillin sulbactam should be reported as resistant when ceftriaxone is resistant or the ESBL test is positive. We don't have clinical data that ampicillin sulbactam works for Enterobacterales having resistance to ceftriaxone or producing an ESBL. Antimicrobial susceptibility testing is not recommended for Burkholderia cepacia. As you know, the CLSI removed the breakpoints and ASU recommendations for this pathogen, and also the UCAS removed the recommendations to test Burkholderia cepacia. So please do not perform AST for Burkholderia. In those cases, infectious diseases consultation is required, and perhaps you can send the isolate for a reference lab, which performs reference blood microdilution. Enterobacterales resistant to any carbapenem tested, ertapenem and or meropenem should be tested for a carbapenemase using phenotypic and or molecular assays. We know that in several institutions in the Caribbean, you are going to receive some methodologies to detect carbapenemases like immunoassays, and in other cases, you are going to receive multiplex PCR to perform syndromic testing directly from positive blood culture bottles or respiratory tract secretions, etc. So it is important to take into account that those mechanisms of resistance, like the carbapenemases, should be ruled out. Enterobacterales intermediate or resistant to ertapenem and or meropenem or positive for carbapenemases should be tested using the VITEK 2 ASTGN403 to provide additional options for treatment. Cefepime should be reported as R or deleted from the report if the isolate is resistant to carbapenems. Nitrofurantoin should not be reported from sources other than urine because it doesn't concentrate, for example, in blood or soft tissue. And fosfomycin should be reported only for E. coli isolates. For Stenotrophomonas maltophilia, take into account that you are supposed to report only trimethoprim sulfa. Okay, so let's move to the VITEK 2 AST402. This card is not new. Several institutions in Latin America and the Caribbean have been using this card. It's just a reminder about what this card contains and how you should use this card with some suggestions, I would say. So this card includes amikacin, ampicillin sulbactam, cefazolin with higher concentration. So in case that you require cefazolin for UTIs, it works, but for systemic infections, you should use 450. Cefepime, ceftazidime, ceftriaxone, ciprofloxacin, ertapenem, the ESBL testing, gentamicin, imipenem, meropenem, piptazo, and tigecycline. So this 402 is indicated for antimicrobial susceptibility testing of gram-negative bacilli for Enterobacterales, Pseudomonas, and Acinetobacter baumannii, isolated from hospital services like ICUs, general wards, or emergency rooms. It could be applied for isolates from urine, blood cultures, body fluids, skin and some tissue, and bone and joint infections. The antibiotic composition of VITEK 402 is suitable for services where carbapenem-resistant isolates are less frequent. Ampicillin sulbactam susceptible results can be used to infer susceptible results to amoxicillin clavulanic acid and can be automated using an expert rule. Lower concentrations for cephalosporins like ceftriaxone, cefepime, ceftazidime, and carbapenems allow for better detection of extended spectrum beta-lactamases like ESBLs and carbapenemases. Which are the recommendations with VITEK 402? So, Enterobacterales resistant to any carbapenem tested, like ertapenem, imipenem, and meropenem, should be tested for a carbapenemase using phenotypic and/or molecular assays. As an exception to this recommendation is Proteus, Providentia, and Morganella that are orally resistant to imipenem because of intrinsic resistance. Enterobacterales, intermediate or resistant to ertapenem and/or meropenem, are positive for carbapenemases should be tested using the VITEK 2403 to provide additional therapeutic options. Tigecycline should not be reported on organisms isolated from the urinary tract. Take into account that tigecycline and as a group, the tetracyclines do not concentrate in the urine. So just avoid reporting this drug for UTIs. Pseudomonas aeruginosa resistant to imipenem, meropenem, and ceftazidime, I would say the three drugs here, should be tested for a carbapenemase using phenotypic and/or molecular assays, despite they are still look susceptible to cefepime or piptazo. When you have resistance to IMI, mero, and ceftazidime, you should perform a carbapenemase test, for instance, a CARBA-MP or other tests that you have available. So what about the 403 card? This is not new. This card is available in some institutions. If you don't have it and you are interested, ask your PharmInpex representative to consider this card. This is a different card. So it includes amikacin, ampicillin sulbactam, aztreonam, cefepime, ceftazidime, ceftazidime evivactam, ceftolozane tazobactam, ciprofloxacin, ertapenem, the ESBL test, gentamicin, imipenem, meropenem, piperacillin tazobactam, and tigecycline. So what about the main features of 403? This card is indicated for antimicrobial susceptibility testing of gram-negative bacilli, for example, Enterobacterales, Pseudomonas aeruginosa, and Acinetobacter baumannii complex from any source isolated from hospital services with high endemicity of multidrug-resistant isolates, especially carbapenem-resistant and/or carbapenemase producers, where testing for new beta-lactamase inhibitor combinations is required. Ceftazidime evivactam is active in class A carbapenemase producers like KPC or GES, and class D carbapenemase producers like OXA48 for both Enterobacterales and Pseudomonas aeruginosa. Ceftolozane tazobactam is active in difficult to treat resistant Pseudomonas aeruginosa, for example, carbapenem resistant, but is inactive against carbapenemase producing either Enterobacterales or Pseudomonas aeruginosa. Oceptolosan is a good drug. It's a very potent anti-pseudomonal drug, but it should not be used or reported as susceptible when you have the confirmation that the isolate is producing a carbapenemase. Astronam is active against Enterobacterales and Pseudomonas producing metalloenzymes, metallocarbapenemases, like VIMs, NDMs, or IMPs. Mostly in your institutions, I know that you have KPCs, but you are going to see some NDMs and perhaps some VIMs or some IMPs, depending about the traffic of tourism, because those tourists and this medical tourism can bring some new enzymes to the islands. So this astronam is useful when combined with Septas avi, with Septacinium avivactam, and susceptible results can be used to infer susceptible results to Astronam avivactam. This is a new drug that is coming to the market probably next year in some territories in the Caribbean. This is a very potent drug covering a lot of enzymes, but unfortunately it's not available so far. But again, I repeat, if you have Klebsiella or E. coli testing susceptible to Astronam, is considered also susceptible to Astronam avivactam. So some recommendations when you use VITEK 2 AST N403. Ceftazidime avivactam should be deleted from the report or reported as R if the isolate produce a metalloenzyme, because metalloenzymes hydrolyze ceftazidime avivactam despite you see those results with a low MIC or susceptible. Astronam should be deleted from the report or reported as R if the isolate produces a KPC. Pseudomonas resistant to carbapenems, IMI, almero, and ceftazidime, ceftazidime itself, should be tested for a carbapenemase using phenotypic and or molecular assays. Here in Pseudomonas at the CLSI, we are looking for methods to rule out carbapenemases, and we think that CARBA-MP could be the first option. Then if positive, you can move to immunoassays or other methods or molecular assays. It depends on the availability that you have at your institution. Ceftolozane tazobactam should be deleted from the report or reported as R if isolates of Enterobacterales or Pseudomonas produces any carbapenemase. And Astronam, ceftazidime avivactam, and ceftolozane tazobactam are not active against Acinetobacter baumannii complex. This is the new VITEK 2 AST 806. This is a very important card that we consider it could be used as a routine card for gram-negatives in some institutions. Obviously, all these card options, you have to discuss it internally, and every single institution should take the decision, should make the decision. But let's discuss about 806. So, this card contains ampicillin, ampicillin sulbactam, cefazolin, cefepime, ceftazidime, ceftriaxone, ciprofloxacin, ertapenem, the ESBL test is included here, gentamicin, levofloxacin, meropenem, nitrofurantoin, piptazo, and trimethoprim sulfa. So which are the main features of the VITEK 2 AST N806? This card is indicated for primary antimicrobial susceptibility testing of community-acquired and hospital-acquired infections caused by Enterobacterales and non-fermentative gram-negative bacilli like Pseudomonas and Acinetobacter baumannii complex. This VITEK 2 ASTGN 806 could be considered as a routine AST card, especially in wards with low endemicity of multi-drug resistant gram-negative bacilli. That's the reason why we consider this could be a routine card. So which are the recommendations when using 806? This card replaces the ASTGN 84, what is in phase out. The 84 is not going to be available in the near future. Enterobacterales intermediate or resistant to ertapenem and or meropenem or positive for carbapenemases should be tested using VITEK 2 XN30 to provide additional therapeutic options. I'm going to show you this in a minute. This is a very brand and good new card. Pseudomonas aeruginosa resistant to ceftazidime and meropenem and or producing a carbapenemase should be tested using the VITEK SN30 to provide additional therapeutic options. And for Stenotrophomonas maltophilia, report only trimethoprim sulfa using the 806. It is important to mention here that in these cases of Stenotrophomonas maltophilia, the current treatment recommended by guidelines is trimethoprim sulfa. However, I think this is going to change in the near future, and Astronam avibactam is becoming the preferred drug, and also cefiderocol that unfortunately is not available in the Caribbean. So here you have a comparison between 806 and GN84. I'm going to show you the deleted, the new, and the update in this card. So this is the old one that is in phase out. It's going to be removed from the market. So in both, you are going to have ampicillin. Ampicillin sulbactam is going to be included in 806. This 806 do not contain amoxicillin clavulanic acid. But when ampicillin sulbactam is susceptible, and this is especially for E. coli, Klebsiella pneumoniae, and Proteus mirabilis, having ampicillin sulbactam susceptible, amoxicillin clavulanic acid should be reported as susceptible without requiring testing itself. I repeat, when ampicillin sulbactam is susceptible, it is considered amoxicillin clavulanic acid susceptible as well. Both cards has cefazolin, but this 806 has lower dilutions for cefazolin compared with the GN84. Look at ceftriaxone. Both cards have ceftriaxone, but if you see here, 806 has lower dilutions. This is very important to detect ESBLs and to predict clinical outcomes. Ceftriaxone in the 806, the new card has lower dilutions and a lower colon range starting at 0.25. Here in GN84, you didn't have ceftazidime. Now you are going to have ceftazidime. What is important, a drug for Pseudomonas. Cefepime is going to be included in the two cards. In 806, it has an update with a lower concentration of cefepime to predict better clinical outcomes. Piptazo, you have it also in the cards. Astronam is not going to be in the 806. You are going to have it in XN30. I'm going to show you in a minute. Ertapenem, it's going to be also in 806, but with a lower dilution to detect better carbapenemases. Meropenem is going to be here also in the 806, the ESBL test as well. Genamicin is going to have, it's going to be included, in the 806 card. Ciprofloxacin now is going to have the lower dilutions that let you match the UCAS and CLSI recommendations for Salmonella. But not only for Salmonella is important. Having lower dilutions for ciprofloxacin, it is important for Shigella also. And in some cases, when the physicians want to de-escalate the patient for Cipro, and obviously having a lower dilution makes them feel more comfortable about switching to an oral options like Cipro. You're going to have levofloxacin, nitrofurantoin. Tetracycline was removed, but take into account that tetracycline does not concentrate well in the urine. Trimethoprim sulfa is included also in 806. So this is a better card. For me, it's an update of what you had in GN84. Also, it has lower dilutions that helps you to have accurate MICs and better prediction of clinical outcomes. So I encourage you with from the bottom of my heart to move to 806. But what happens if I have a multi-drug-resistant pathogens whenever I test, for instance, 806? So now you are going to have this new card XN30. In this card XN30, you are going to have available amikacin, amoxicillin clavulanic acid, astronam, cefotaxime, cepodoxime, ceftazidime avibactam, ceftolozane tazobactam, doxycycline, eravacycline, that is important for Acinetobacter. I don't think this drug is available in the Caribbean so far, but you should ask. Imipenem relebactam, what is a new drug. This is a potent new inhibitor. This is a second generation of avibactam, and this imipenem relebactam is important for Pseudomonas and KPC producing isolates, mostly the ones which are resistant already to ceftazidime avibactam. So imipenem relebactam is an important therapeutic option. Meropenem vaporbactam, this is also a boronic acid derivative. And vaporbactam is a potent inhibitor of KPCs. Not sure if this drug is available in some islands. However, you should ask because this could be an important alternative. Also, we are going to have here minocycline, important also for Acinetobacter, and polymyxin B. This is very interesting for me because polymyxin B is not my preferred drug. I should say that every time that we in Colombia and other countries have or we face a carbapenem resistance to the Pseudomonas or Acinetobacter, physicians use other drugs. We try to look other alternatives beside polymyxins or colistin, I mean. But in some institutions which do not have alternatives or having or facing Acinetobacter outbreaks or severe infections caused by those pathogens, polymyxin and colistin could be a therapeutic option. Also, this card has tigecycline and tobramycin. That is an aminoglycoside very potent against Pseudomonas and is available in some Caribbean territories. So this is a new card. It is important to mention that to perform XN30, you are required to perform 806. You are not allowed to perform XN30 without or testing 806 first, because this is an extension card. If you use other cards like 402 or 450, you are going to have 403 as an alternative to test for additional drugs. So recommendations when using VITEK 2 AST XN30. Meropenem vaporbactam should not be reported in Pseudomonas aeruginosa because it's not very active and it doesn't provide any advantage against meropenem itself. Imipenem relebactam should not be reported in Proteus, Morganella, and Providencia. Ceftazidime avibactam, imipenem relebactam, and meropenem vaporbactam should be deleted from the report or reported as R if the isolate produces a metalloenzyme like NDM or VIM or IMP. Astronam should be deleted from the report or reported as R if the isolate produces a KPC. Ceftolozane tazobactam should be deleted from the report or reported as R if isolates of Enterobacterialis or Pseudomonas aeruginosa produces any carbapenemase. Take into account that Astronam, imipenem relebactam, meropenem vaporbactam, ceftazidime avibactam, and ceftolozane tazobactam are not active against Acinetobacter baumannii complex. Polymyxin B results, either intermediate or resistant, can be extrapolated to colistin because in some islands you don't have polymyxin B itself. You have colistin, but testing polymyxin B is a surrogate for colistin and you can report them exchangeably. Polymyxin B should be reported as colistin on organisms isolated from the urinary tract because from the urinary tract, polymyxin B does not concentrate well. So the drug that concentrates well in the urine is colistin. But as I already mentioned, polymyxin B and colistin can be reported exchangeably. And polymyxin B should not be reported in Proteus, Morganella, and Providencia because of intrinsic resistance. So we are going to navigate this workflow depending on which cards you are going to use as primary cards and which other you can use for additional cards to provide therapeutic options for multi-drug resistant gram-negatives. So let's start. With Enterobacterialis other than Salmonella and Shigella, the primary cards could be VITEK 2 GN 450 or VITEK 2GN402. Take into account that Enterobacterales is intermediate or resistant to any carbapenem except imipenem for Proteus, Margandella, and Providencia. Test for carbapenemases using phenotypic and or molecular assays, and use VITEK 2 ASTGN403 for additional antibiotics. For Salmonella and Shigella, the primary cars will be the VITEK 2GN450 and the VITEK 2GN806. For isolate resistant to ceftriaxone, take into account report ertapenem. For Pseudomonas aeruginosa, you can have several options for primary cards. The VITEK 2GN402 and 403, or the VITEK 2GN806. And take into account that Pseudomonas resistant to ceftazidime and carbapenems. You should, if you can, test for carbapenemases using phenotypic and or molecular assays. If VITEK 2GN806 was used as primary card, consider VITEK 2SN30 for additional antibiotics. If VITEK 2 AST402 was used as primary card, use ASTN403 for additional antibiotics. What about Acinetobacter baumannii complex? The primary cards will be 402, 403, or GN806. Acinetobacter baumannii complex, which are resistant to ampicillin, sulbactam, and meropenem. You could test for carbapenemases using phenotypic and or molecular assays. And if VITEK 2 GN806 was used as primary card, consider VITEK 2 SN30 for additional drugs. And for Stenotrophomonas maltophilia, the primary cards will be GN450 or GN806, and to report only trimethoprim sulfa. Obviously, dear friends, this is only a suggestion. Each institution should do their internal discussion and make a decision about what would fit best in your local treatment guidelines and antimicrobial stewardship approaches. So this is coming soon. We are going to have a booklet, Cards for Antimicrobial Susceptibility Testing in the Caribbean, Microbiology with Reliability. I made this booklet along with Pharmipex and Biomeru. This is going to include the recommendations and some suggestions for you to implement these cards. So we are finalizing the editing of this document, and you are going to receive this very soon. So please stay tuned. In summary, the new VITEK 2 cards provide AST for gram-negative pathogens in several settings. It is important to discuss in each institution the availability of the drugs in the hospital formulary, antimicrobial treatment guidelines, and stewardship practices. Each institution should be free to decide what cards to test for clinical purposes. We are not looking to obligate nobody to choose a particular card. It's your decision. But Pharmipet, supported by Biomeru, is going to provide the alternatives as were discussed today. What we can do, if Pharmipex and Biomeru agrees, we can have like a training session about results on how to interpret the mechanisms of resistance about what we have been facing in the Caribbean. Remember that it's important to detect the mechanism of resistance behind those pathogens, because it's the information that will help physicians to make a well-informed decision. And please contact your Pharmipest representative to implement these cards in your instrument, because it might require some updates, some software updates, or it also depends on your needs to implement these cards. But Pharmipex will provide help about this. So thank you very much. This is what we got for today. I'm going to open the floor for questions and discussion. I'm going to switch back to Emily to lead this. Thank you very much.
Thank you, Professor Esparza, for this comprehensive and insightful presentation. You provided a detailed overview of the VITEK 2 gram-negative card, including composition, recommendation for use, etc. I know it was a lot of valuable information to absorb, and we truly appreciate the clarity and depth of your explanation. Although it may take some time for all this knowledge to fully settle, I would like to encourage everyone. If you have any urgent or pressing question now is the perfect moment to ask. This is a unique opportunity to have the direct access to our Professor Esparza expertise. Um, so please text your question in the Q&A box or in the chat box. Professor Esparza is able to see the Q&A box. So he will make sure to address all your questions. So please feel free and ask the question. We have
Okay.
received already in the chat
It
box and in the Q&A session if we are able to send the presentation and the recording of this webinar. Yes, we are able to do so, but please let us use the time now to discuss all the questions we have. Um, Professor Esparza, can you see the Q&A box also?
Yes, I can. So Stacy
Yes.
asked us about the recording and the PPT slides. And yes, we are going to share the recording. I know, as was mentioned by Emily, that this is a big amount of information and recommendations just for 40 minutes, but I'm going to suggest, if you will, that we can have some examples, training with cases in a separate webinar that we can, in the way that you are implementing these cards, getting used to the results and the phenotypes that you are going to see to help you incorporate those concepts in your daily practice. So keep in touch. We are going to do so to keep training you in the use of these cards. So no worries at all.
Okay. Thank you, Professor Esparza. In the meantime, I'm looking if we get any questions. Not really. Excellent presentation. Yes, it was indeed an excellent presentation.
Thank you.
I wait for a bit more to see. We got one question. Herman, can you see it?
In the chat box?
Yes, in the chat.
Okay, so Dr. Blades.
From
Hi, Dr.
Dr.
Blades.
Blades.
Yes,
Yes.
from our dear friend Edmund Blades. So Dr. Blades is asking us, since there is no longer antibiotic recommended for Burkholderia cepacia, what labs should be recommended? So what we as the CLSI are recommending is to include a footnote below the identification of the pathogens, below the Burkholderia cepacia, that AST methods are not accurate, and it applies for all automated systems from all brands. So the patient should undergo infectious diseases consultation to find out what will be the best treatment. Because the problem, Dr. Blaze, with the AST methods for Burkholderia cepacia is that it can provide false susceptibility or false resistance. When you have a meropenem susceptible, you don't know if it's really susceptible or is a resistant pathogen and could mislead the treatment and obviously can cause a bad outcome. So this is what we are doing. Something else that you can do is perform the reference broth, but I don't think in the islands it's available. I should ask to my colleagues at CARFA, but I don't think CARFA is doing broad microdeletion for Burkholderia cepacia so far. I should ask them. But this is the other thing that you can do, and you can report the MIC only. But you have to do reference broth with the frozen panels, with the M07 or ISO 20766. But not with the commercial device manufacturers from any brand because it's not performing well. The good news is that Burkholderia cepacia is not the first pathogen in the institutions, and sometimes it may mean colonization or contamination. It could be contamination in some cases. So take this into account. And the CLSI is trying to provide new guidelines, and also the Infectious Diseases Society of America is going to publish some recommendations for Burkholderia and other non-fermentative gram-negative pathogens.
Okay, thank you. Since we are coming to the end of this session,
there are some questions in the Q&A. So,
sorry, can you see?
Yes, Dr. Van der Roost says, is there a possibility to incorporate carbapenemase resistant detections just like the SBLs into new cards? No, sir. At this point, we do not have carbapenemase detection for those cards. The problem with carbapenemase detection, there are some commercial devices that actually have detection of carbapenemases using inhibitors. What is the problem? The problem, Dr. Van der Roos, is that it is causing false susceptibles and false resistant when you have more than one enzyme. That's the problem. That's the reason why it really works for the US, but when those devices came to the market here in South America, as we have KPC plus NDM, NDN plus whatever, VIMs like Pseudomonas, VIMs like KPCs, we have an addition of several enzymes detected simultaneously. Those tests perform really poorly. So unfortunately, we don't have this. We recommend to perform ancillary tests like immunoassays or PCR according to your availability. Also, our colleagues at Argentina have some inhibitor-based synergy testing that could be helpful, but not at this point. Beside the ESBL, any other test is going to be included. And what I feel about the ESBL test is that unfortunately is very unspecific right now, because we have KPCs providing false positive KPC results and some ESBLs false susceptible when you have a KPC, and some ESBLs not detected by the ESBL method, the manual anti-automated. So I think that in the future, the ESBL test is also having poor accuracy. So sorry about that. Is there any? Okay, Victoria Labs, and I'm gonna send my greetings to Victoria. I think it's in Trinidad. Am I right?
Yes.
San Fernando in Trinidad. Okay.
Yes,
so,
exactly.
Okay, I recall it. So why can't the GN450 be used for Pseudomonas? You can use it, but the problem is that it doesn't incorporate all the drugs that you need for Pseudomonas. If you perform outpatient isolates, or community isolates, mostly those Pseudomonas are going to receive ciprofloxacin because it's the only oral drug. But the problem is that if ciprofloxacin is resistant, and this is a big deal, an important infection, you have to hospitalize those patients, and you need all the IV drugs. So that's the reason why I recommend you respectfully that you can use, instead of 450 for Pseudomonas, you may use 402, 403, or even 806. So I ask you to please contact Farming Pets, and we can provide some suggestions. If you will, we can have a video call, and discuss the options. But my recommendation said that you are not supposed to use 450 for Pseudomonas. Okay, thank you. There is something else?
Hi, good morning. There is an additional question, not in the Q&A, but in the chat box. Maybe we can check those out.
Okay, I found it. Okay, hold on 1 second.
Yes.
It
is from.
Thank you. Okay, so it says the GN84 is used for Pseudomonas when they get a susceptibility for carbapenem resistant. Are you saying that the new car we will get a better susceptibility result? Also, can we still use the GN84, or do we have to switch right away? Okay, I'm gonna answer the first part of the question, and I'm going to switch to my colleagues at Formipex to answer about the GN48 availability and the phase-out process. Okay, so for Sam's laboratory, and I'm going to send a greeting for them. The 806, it's a better car for Pseudomonas and Enterobacterales because it has lower dilutions, is more accurate, and it incorporates the drug that you need for Pseudomonas aeruginosa. In case that you have a carbapenem-resistant and ceftazidime-resistant Pseudomonas, you can look for carbapenemases and you can use XN30 for additional drugs. It depends, obviously, on the availability of those drugs at your institutions that you are providing the service. If you don't have the new drugs, probably you're not required to test an additional card, just the 806. But yes, my suggestion in the way that you can is to switch or to move from a GN 84 to 806 because it's more accurate for susceptibility testing. I'm going to switch to my colleagues at Pharmipex to answer.
Yes, let's switch to Shirlene Cecilia from the implementation team.
Yes, good morning. Good morning, everyone. Yes, as for the availability and the switch over to the new card, the 806, as German just mentioned, the intention is to switch as soon as possible. Of course, you have received recently some 80 GN84 cards, but we are going to have the 806, the N806 as soon as possible available for you to switch over right away and start using this new card.
Thank you very much, Shirlene. Is there any other question? Ask whatever you want. Okay, Tara Lewis says, we recently switched to 806. In your suggested algorithm, it is not listed as the primary card for Enterobacterales. Oh, I think I did. Just let me share my screen again. Just hold on 1 second. There you go. Probably I moved too fast. Sorry about that. Okay, so here you can have 806 for Salmonella and Shigella, and you can have also for Enterobacterales 806. The point is that if you are going to use 806 for Enterobacterales, you can use XN30 to test additional drugs. But if you don't start with 806 and if you start with GN450 or 402, you are supposed to use 403 for additional drugs. We are going to provide like a booklet to do so. But yes, Tara, you can use 806 also as primary card for Enterobacterales. It's suitable, as you mentioned. Is a good card, and if you don't have the new drugs, or if you have, for example, pathogens that are not quite resistant to carbapenems, you can just use 806. So no problem. Yes, you can use as primary card.
Emily, I think you're muted.
Yes, I was just checking also the Q&A box. Can you switch also? Because we have one from Ian van der Roost from Curaçao.
I can see it.
Okay, I will read it out for you. Okay. Is there a possibility to incorporate carbapenemase?
Oh, I answered already.
Yes.
Oh, yeah, I answered already.
Okay.
No
Okay. Yes. Then we are okay. I think let me check more.
I think I can answer the other one from Ian. He just asked if the N447 that was recently introduced, launched for the Dutch Caribbean, that it will be obsolete in the near future and replaced for the 806. No, Ian, no, this is not going to be the case because this is also one of the new cards with the new drug version, with the performance, with all the changes, and based on the guidelines for EUCAS and also CLSI guidelines. So it will not be replaced in the near future.
Yes.
Okay. So If there is no any other question, I would like to thank my colleagues at Farming Pest, Emily, Shirlene, Tishani, Saritza, Harry, and everyone in this room, and also the international division from BioMerieux. I would love to see you again, where we can train with some practical cases when you are implementing these new cards. And I would love to visit you again in the near future. So thank you very much.
Okay, thank you, Herman. Also, on behalf of Farm Impacts and BioMerieux, we are truly grateful for the time you have given us and your contribution to improving clinical microbiology practices throughout the Caribbean. At Farm Impacts, we are committed not only to providing innovative diagnostic solutions, but also to supporting our customer every step of the way, from implementation to training and ongoing technical assistance. If you have any question after today's session or need support with integrating the VITEK 2 gram-negative cards into your workflow, please don't hesitate to reach out to the team. You can contact Shirlene Cecilia from the implementation team or our application specialist, Saritza Martina. They are always available to help you with any inquiries or practical guidance you may need. And to all of you who joined us today, thank you. Thank you for your presence, your engagement, and your dedication to advance this healthcare in our region and in our Caribbean. We hope today's session was meaningful and that it inspires continued learning, collaboration, and innovation in your laboratories. Thank you again. We are looking forward to staying connected with you. I want also to encourage you to please fill in the evaluation form with when you fill in this evaluation form, you will get also a certification of participant at this webinar. Thank you again and have a nice day. Bye.